If you made it to this video, you probably came here because you're trying to solve the mystery that is my in-class exercise. So my in-class exercise comes from an exercise I did many, many years ago when I was teaching a biology class that met once a week. It was a half a semester. I think it was a 12th week. It was once a week on a Thursday. And we met from 6 o'clock at night in person until 9 o'clock at night. And when the first day I came to the classroom, I discovered that I had a room without a computer. And I thought, I'm going to die. But we made it through 3 hours of teaching. And at the end of 3 hours, I still had students that were surprisingly awake. So by the next lesson, I requested a room with a computer. And about 5 minutes into the lecture, my students were asleep. And so that particular group, I did a lot of activities with them. So they were pretty used to, me trying to keep them awake for 3 hours. And so one of the activities I did with them was this photosynthesis cellular respiration in-class activity. I came in one day and I told them to put their books away, and I handed them a piece of paper that had this picture on it. Basically, I had photocopied right from the book, because this was the olden days. I'd photocopied from the book this image, and I deleted the labels. And they had this one, and then this one. And then I asked them to label it. And they looked at me like I was crazy. And I said, no, seriously. Think about what you already know. Because there are things you know about photosynthesis and cellular respiration. You know things about, and what one of the things about learning is you have to connect what you know with what's new. And your brain functions by making connections. It's literal neurons making literal connections, literal pathways from one piece of knowledge to another piece of knowledge. We learn by association. So what I want to do is I want to try to connect what you already know to what you're just learning here in this chapter. And it's interesting because it matters. This is biology. You're a living, breathing human, last time I checked. So you do this. This is a thing. Many of us are interested in weight loss, weight gain. All of this matters in weight loss, weight gain. It matters in exercise. It matters in muscle building. It matters in all of those things. If you're interested in any of those things, photosynthesis and cellular respiration are your friends. Learn about them. It gives you a deeper understanding of how some of these processes work. So let's play this and see how far we can get through this. And as I'm going to the PowerPoint and through this, now start thinking about and pause the video. I'm going to ask you to pause the video multiple times. Think about what you know. What are you breathing in? What do you need to survive? What are you breathing out? What do plants provide us with? What do plants inhale? What do plants need to survive? What's energy? Where does it come from? Where does all energy come from? So those are the kinds of things that I want you to think about. Okay. So one of the things I want to share with you is this Google Doc. It's a presentation that you can actually use to actively do things. You can find the presentation at this tiny URL. So just type this into your browser. It'll give you access to this presentation. You'll have to download it. So it'll come up. You won't be able to do anything with it at that moment in time. And you can save it. So you basically make a copy of your own. And once you have your own copy, what you can then do is you literally move these things around. And so let's see if we can label some of these things. And as we talk about, what do we already know? So first things first, if you've looked at your chapter, if you haven't looked at your chapter yet, you probably should. What we're talking about plants, right? Plants and animals, and we're talking about the transfer of energy from one system to another. We're basically talking about the um, the thing that powers spaceship Earth, for a lack of a better word. And the thing that powers spaceship Earth, if I were, you know, to push you to give me an answer to that, um, and those are the choices I gave you, you'd probably be able to come up with the fact that spaceship Earth is probably powered with sunlight, right? And so look at these. Go to the tiny URL. There's the tiny URL. Download this thing for yourself. Let's see if you can figure out where the labels go on this slide. Based on the things that you already know. So what we're talking about here, if you haven't looked at the chapter yet at all, if you look at this picture, and students don't often look at pictures. Look at pictures in biology textbooks. There's a green thing, and there's a ray, you know, and then there's a brown thing, and there's a spark, and then there's something radiating off. There seems to be a cycle, and then there seems to be something passing through. Just looking at it. And now I'm going to tell you, if you haven't looked at the chapter yet, that this cycle is what happens on Spaceship Earth. And Spaceship Earth is powered by one thing. So the first thing you want to label is probably the thing that powers spaceship Earth, which is sunlight. Now the thing that gets lost, that's not utilized, that's heat energy. You may not have thought about that. Now, photosynthesis in chloroplasts is happening somewhere. Cellular respiration in mitochondria is happening somewhere. One of these is ATP. O2, that's oxygen. Somewhere oxygen is being produced, and somewhere oxygen is being incorporated. So pause this video here for a second and see if you can label this. I'll wait. Did you do it? All right, let me pause it. If you need some more time, I'll go through it now. So the green stuff, that's where photosynthesis is happening, right? So photosynthesis is happening in chloroplasts right there. And what's the outcome of photosynthesis? What do plants produce for us? Hopefully, we know that we need trees and stuff for the oxygen, right? So that goes there. Now, we breathe in oxygen and we breathe out CO2, right? And so, how convenient. We breathe this out, and then plants actually breathe in CO2. So, this is a cycle, right? Um, Sunlight is captured by the plants in the form of organic molecules. They pass these organic molecules on to mitochondria, who then utilize them to produce ATP. And ATP is used to power most of the cellular work, which leaves us with the last little label here, which is cellular respiration mitochondria. That's what's happening to produce the CO2 and the water and the ATP. So these are the waste products of the ATP production. Um, that's the part of that's the great big overview of what's happening here. Now, one of the things that students struggle with, and I actually didn't ask about it, and I don't ask about it in the in-class exercise, but since we're talking about it, let's talk about oxidation reduction reactions. Reduction reactions suffer from a very bad um, naming nomenclature, basically, naming system. But the basic gist of it is you have to remember that oxygen is a bully. And oxygen is highly, highly electronegative, which is basically another chemistry term for bully. And what oxygen has 6 electrons on its outer shell. It wants 8. And it is very good at pulling electrons from other places to fill that shell. Now, in biology, most things in biology happen in aqueous systems. So anytime electrons move, protons move, anytime anything moves, there's always a couple of hydrogens that are running around to go along with that. Because everything happens in water, and anytime you have a charge somewhere, a hydrogen, fixes the problems with whatever charges happen to be. Electrons happen to free floating around. Um, so what you have here in this situation, you have one of these. You have an oxidation reduction reaction where the fact that the reaction happens is because there's a pull on the part of somebody trying to get electrons closer to themselves. Um, and so the oxidation reduction reaction usually means that oxygen pulls electrons to itself. And when oxygen pulls electrons to itself, the other thing is said to be oxidized. Oxygen itself becomes reduced. So as it's taking on these electrons, it's becoming reduced. So this thing here, oxygen going to water, that's the reduction stage. And they always pair together. This one's the oxidation. In the next one, we've got the opposite way. We have water going to just to oxygen. That is a loss of electrons, and a loss of electrons is an oxidation. Um, and that's one of those things that's oftentimes students are struggling with. Remember, um, loss of electrons, oil rig, oxidation is loss, reduction is gain. So this oxidation here is a loss of electrons. And in this case, it looks like a loss of a hydrogen, but it's really a loss of an electron. It only is a loss of a hydrogen because it's within an aqueous solution. So far, so good, I hope. So this is actually probably the toughest part. The rest of it, most of this you know. So let's look at this one and see if you can label these. Now also look at the symbols. The symbols are the same between the different slides. So once you figured out what something is on one slide, it's going to be the same thing on the next slide. So um, now this thing, what we're talking about here, this is a chloroplast. Where does a chloroplast reside? It resides inside of a cell, right? So it's not its own cell. It's a little piece, a little bacterial size, little form or organism that is inside of a eukaryotic cell. Um, and so this little baby here is a chloroplast. Now, chloroplasts have structures inside of them that you may want to know about, may know about. You should learn those structures. Those are the thylakoids. The thylakoids, they are the one that capture the energy, right? The energy that's like overall the thing that powers spaceship Earth. And so this energy also is called light. What else do you already know about photosynthesis? You know about photosynthesis that plants produce oxygen, right? So here are the products. Here are the inputs of photosynthesis. Now, we've already looked at this reaction earlier, right? We had this reaction earlier. And this reaction tells us, actually, we had this reaction here. We said CO2 and water get turned into organic molecules and oxygen. CO2 and water get turned into organic molecules and oxygen. CO2 and water get turned into organic molecules and oxygen. So which one of these up here, 4 and 5, is the CO2 and which one is the water? Now these symbols are probably labeled the way they are because of, they sort of kind of make sense. And so if I'd have to guess, I'd stick this CO2 and the water right there. Those are the inputs. Now let's think about the outcomes. Everything is chemistry. So your outcome is going to be oxygen and sugar. Sugar contains carbon. That's the whole point. They're carbon compounds. So which one of those do you think is probably the sugar? Probably the one that has the carbon input, right? Because there's where the carbon comes in and the carbon gets utilized over here. So sugar goes here, oxygen goes there. Now we're getting somewhere here, right? All right, what else do we know? Well, light, the light reactions, those happen over here. And those happen in the thylakoids within those green structures that are inside of the plant. The reason why they happen inside of those green structures is because the green part of the green structure is actually the thing that makes the thing happen, that allows the reaction to happen to start with. Um, Now this reaction over here happens in the stroma, and that's called the Calvin cycle. It used to be called the dark reaction or the light independent reaction. We now call it the Calvin cycle. Now we've cleaned up pretty well most of them. We got a couple of them left. The whole point of this reaction right here, well, the whole point of the overall reaction is the storage of energy, the long-term storage of energy. What you're starting out with is you're starting with a photon. That is a very fleeting kind of energy, right? It's ding and it's gone. You turn the lights off, the energy's gone. So what happens in the thylakoids is that this energy from light is incorporated into something that allows the plant or whatever organism does the photosynthesis to hold on to the light, to hold on to that energy for just a little bit longer. And it does that in form of the energy currency ATP, which is what that explosion looking thing always is. ATP cycles between ADP, adenosine diphosphate, two of them, and a phosphate, and ATP. So this goes around and around, and what happens here is this is basically an uncharged battery, and it's charged up here. So then this energy then gets fed into the Calvin cycle, and it is then incorporated in the Calvin cycle into the sugar, because you have a very, very fleeting short-term type of energy that gets turned into an energy currency that holds just a hair longer, like the difference between a photon and I don't know, firecracker. And then here, this energy in that little firecracker is stored into sugar that you can literally stick in a shelf. And that's what plants are trying to use it for too. The other raw material that the Calvin cycle requires in order to function and to be able to be recycled over and over again is it needs electrons. And so these electrons are carried in a little suitcase by NAD. Now the question is, does NADP plus or NADPH go down here? And remember when I told you that everything in biology always, if you have some extra electrons, you probably have a couple of extra hydrogens floating around or something. So the NADPH, the ones with the extra hydrogens, the extra protons that are hanging out there because of the extra electrons that are hanging out. And then here you've got uh, the other one, NADP plus. And there we go. This is labeled. Now, if you've just watched me do this, go back to the tiny URL and see if you can follow it through. Can you do those labels just exactly like what I just did? And can you think through it yourself? Now, the next one we're going to do is actually a little more scary looking than that one. That's the mitochondrial one for cellular respiration. And But again, by now you already know some of the symbols, right? And so since you already know some of the symbols, maybe it's going to be a little less scary. So this is what's happening in the mitochondria, right? And we call this thing cellular respiration. So this is now this part over here, right? So where we had, for the photosynthesis part, we had CO2 and water coming in and sugar and oxygen going out. Now in the mitochondria, we have sugar and oxygen going in, and we have CO2 and water coming out. Now listen to what I just said. We have sugar and oxygen coming in. What drives your body, what fuels your body for most of what your body does is sugar, glucose. That's the fuel. Everything you eat, everything you consume, in the end, if it's used to fuel your body, is turned to glucose first. Glucose is the quickest, the fastest way to get energy to you. The closer you are to sugar, the closer it is to fuel. The problem with your body is you don't want to necessarily go to fuel so fast. But if you need fuel fast and you need it now, you want glucose straight up. Now, when we're talking about calories in, calories out, and glucose is also where it's at. Everything that comes in, there's no such thing. When we're talking about empty calories, they're not empty. They still have the nutrition, right? The emptiness is they don't have anything else. They just have the nutrition. They don't have the vitamins. But what they do have is energy. So make sure you understand what conversations you are having here. But let's look at what mitochondria do for us. First, let's label that this is happening in the mitochondrium right over there. Now, what all do we already know? We can already label some of the symbols, right? That we've already encountered once before, right? Those little starburst-looking little things, those were ATP. So that is, again, that's the energy currency that's in your body that drives everything. It drives all the labor in your body, everything that needs energy input in order to function, and a lot of your enzyme tools do. Those require ATP, but ATP functions only on the very short distance. So ATP, like, it needs to be produced right where it's needed, so it can't be transported over long distances. It's like little firecrackers that are like right there, functioning right there. um, so that's your ATP. What else do we know? Well, this was the water, right? We've already got that. Here's our oxygen. So our oxygen is converted into water. And remember, we talked about this when we talked about the oxidation reduction reactions that are happening here. Your oxygen is turned into water. This is part of that reaction that's happening there. So oxygen goes to water. And then, so as you're labeling these, think about what's happening here. So the CO2 is unloaded over here. This is where the sugar is being taken apart, right? So what comes into the equation is sugar, glucose. And glucose, it needs a little bit of tweaking before it can actually be shuttled into the mitochondrium itself. This tweaking of the glucose happens inside of the cell. So glucose enters the cell, and then inside of the cell is tweaked by a process called glycolysis, which is a very, very old process that predates the existence of mitochondria. So before there were mitochondria, cells took glucose and turned it into pyruvate, and they got a little bit of ATP by doing that. And so they kind of, did their thing. But once mitochondria were around, then mitochondria went, ooh, we can extract way more energy from the same glucose. And the way they do this is the first step inside of the mitochondria. Um, it processes the pyruvate, and in the pyruvate is being oxidized, and as it does so, it produces there are electrons that are being shuttled, um, that are being transferred over, that are being carried by NADH, just as before. And this time there's a secondary one, FADH2, that's also available there. And those electrons, those carriers are there to simply shuttle electrons, um, or because we're in an aqueous solution, um, uh, protons, that kind of thing, into this magic that's happening over there, where the majority of the ATP is produced. And the majority of the ATP is produced in oxidative phosphorylation. And we're going to look at oxidative phosphorylation because it's magic. This is where we're basically taking so what's happening here in the mitochondrium, we have the long-term storage of glucose, and what we're having to do now, we're going to take this molecule apart, and extract the energy that is contained in this molecule. Because earlier we, took a bunch of CO2 molecules. Here, we took a bunch of CO2 molecules and stuffed them together into a carbon chain, and then there was energy contained in this carbon chain, and now we're going to extract that energy from this carbon chain. So we got the carbon chain, now we're going to extract the energy, and we're going to turn this thing back into CO2. And so that's where the CO2 comes in. There's a little bit of energy that can be extracted right here, but the big bang for the buck comes in the shuttling of the electrons into the oxidative phosphorylation, where then oxygen is the thing that drives the whole thing. So oxygen is the fuel. It's the fire that drives. So glucose is the fuel that drives the body, but oxygen is the fuel that drives the mitochondria. It is the final electron acceptor. So it's the thing that does the pulling of the electrons that drives the whole thing. I hope I haven't lost you yet, because we're going down another level of molecules. Because here we are at the actual membranes where these things are happening. And the first membrane we're looking at here is the chloroplast. So this little baby is your chloroplast. And your chloroplast is has a membrane. That's important. All of these things are happening only because there's a membrane that exists. And one of the things that's important to look at for both chloroplasts and mitochondria is that both chloroplasts and mitochondria have their own ways of increasing the surface area of the membrane that they have, because the function that they have is dependent upon how much membrane area they have, because all the things that are happening in the membrane. And so if you have more membrane, you can stick more stuff in that membrane. And so mitochondria do this by having these invaginations. Chloroplasts do it by having the thylakoid stacks. So what you're looking at here is a thylakoid stack. And the reason why the thylakoid stacks are green is because you have, lo and behold, you've got these little um, pigments in there. And these little pigments are the reason why this whole thing is working at all. But so here are the thylakoid membranes. So this is what those are, what those are labeled as. And what else do we know right off the bat? So again, the labels are the same as the labels before, right? The symbols are the same as the symbols before. So some of these things you already know. Um, and even though we're further down in the, looking deeper into the electrons, the reactions are still the same, right? On our chloroplasts, we said we've got water coming in, oxygen going out. And this part of the reaction is the water coming in, oxygen coming out correction, because this part here is only this part here. So what we're talking about here is water coming in, oxygen going out, but we're shuttling both the ATP elsewhere, and we're shuttling a couple of electrons elsewhere into the Calvin cycle that happens over there. So over there, this is where your Calvin cycle is. So your Calvin cycle, there's ATP being provided to the Calvin cycle and electrons. So really, this is just a setup so that the Calvin cycle can then incorporate both of those things and do the long-term storage of the glucose. But that Calvin cycle is a different story for a different day. What this exercise is about is more about the compare-contrast between photosynthesis and cellular respiration. Because what I want you to see is that chloroplasts and a lot of things in nature are variations on a theme. Similar things are repackaged. Nature is a tinkerer, not an engineer. So different things are repackaged in different ways and then utilized in different ways over again. And so this is one of those, because you have um, two structures inside of cells, inside of eukaryotic cells, that appear to do opposing things, right? Because they're like part of the same cycle, but they do like this opposite-y kind of thing, right? It looks like this one provides what the other one needs, and it looks like it's an opposite kind of thing, but they're doing it. It's not an opposite thing, because what you have here is what's happening here is actually very similar to what's happening in mitochondria. Because what mitochondria do, they produce ATP that is being utilized, right? Well, same here. They produce ATP that is being utilized, and they produce the electrons with the NADPH. But let's get back to this little labeling exercise before I get completely lost. To pull us back, so hydrogen, there's our little hydrogen down here, and then our little oxygen, and it's only a half of one, so that's one oxygen molecule per water. And what the water is doing here, and I just want to point that out, the water is providing electrons. Because remember, this is again an oxidation reduction reaction. But what the water is providing here, it's providing electrons that are fueling this thing, which is the electron transport chain. So, electron transport chain, there is our electron transport chain. And what the electron transport chain does is it fuels the production of electrons over here that have to be shuttled to the Calvin cycle. So remember how electrons were shuttled to the Calvin cycle? They were shuttled to the Calvin cycle with NADPH, and NADPH shuttled. It cycled between NADP and NADP plus and NADPH, which is the loaded version of the suitcase, which shuttles to the Calvin cycle. Now these two here, photosystem 1 and photosystem 2, those are the photosystems that capture the light and convert the light into something that basically, something fleeting, that we turn it into some chemical something we can then use. And so this green stuff is called chlorophyll, and chlorophyll is a pigment that has an electron in it, and this electron, when a photon hits it, the electron reaches a higher level of energy. And when it reaches this higher level of energy, if there's nothing there to capture it, this higher level of energy, this little piece of energy that just got incorporated in this electron, the electron falls back down and it does this thing called fluorescence. What these guys do, what the pigments do, they capture this energy. So basically the light lifts up the electron and something in the like to a higher electronic state, but you can think of it as just it lifts it up in the air. Um, and something in the chlorophyll grabs it from there and captures that energy. And then that electron gets passed down the electron transport chain. Another hit of light hits it in this second photosystem, and because scientists are scientists, photosystem 1 is first and photosystem 2 is second. I mean, the other way around. Photosystem 2 is first and photosystem 1 is second. So another hit of light gets the electron then actually charged enough so it can be loaded up on the NADP plus in order to be shuttled off to the Calvin cycle. Now, the outcome of all of this, and I guess I need to put the light here and the light there. The outcome of all of this production over here, where we're shuttling these electrons to the Kelvin cycle, is all from the breakdown of the water over here, from the splitting of the water that's happening. The electrons got shuttled off, but what was left behind is all kinds of protons. Within this membrane is a whole collection of protons, and with every little light molecule and every little water molecule, there's another proton that gets stuffed inside of this membrane. And the more and more of them are in there, the more they want out. Now there's only one way out. And the one way out is through this little molecule called the ATP synthase. And the ATP synthase is a pretty magical little thing. It's a turbine. It's a literal turbine. So you've seen waterfalls, that kind of stuff before, right? And you've seen like mills that, hopefully you've seen mills that run on like a water mill. There's water running over it, and then that water running over, it drives the mill around in a circle, and that is we can then use that to drive labor beyond that. This labor is you're capturing this energy. And that's the same thing that's happening here. So the ATP synthase, these protons would really, really like to leave. And so this is a proton gradient. And this gradient, because they're flowing out, just like water would be flowing out that's under pressure, they're flowing out and they're pushing this thing around in a literal circle. This literally turns. It's a molecule that literally turns, and the turning motion causes ADP plus phosphate to be turned into ATP, which is the little firecracker again, which holds on to energy just a little longer, just long enough to be passed on into the Calvin cycle, where it then is utilized to grab the CO2 from the air, or grab the CO2 that was grabbed from the air, and stuff it together and put energy in it. So that's the chloroplast. A mitochondrium looks very different from a chloroplast, right? We could possibly agree with that, right? And we just looked at the very depth of a chloroplast. Now look at the very depth of a mitochondrium on the next slide. Anything look super familiar here? This little friend here? That little friend here? They sort of look alike, right? And they are. They're very closely related to each other. Both of them are an ATP synthase. Both of them are driven by proton gradients. In both cases, you've got ATP production. What you have here, though, is happening in mitochondria. But you've got ADP going to ATP. You have the production of water. Here's your oxygen. That's the driving force that pulls the whole thing through. Here's your electron transport chain. Here's your, now can you label it even without the labels now? Here's your NADPH, right? Here are your protons that have been pumped through into the membrane, putting up this proton gradient, which then drives the ATP. And this ATP then drives all the processes. So hopefully by going through this and spending a little time just looking at these things, you can kind of compare and contrast, and you can kind of see how these things go together. And it's themes on two different organisms. One used to be a free-floating organism, became a chloroplast. And it happened to have, a little mechanism that captures light and produces using a proton gradient, um, capturing energy in ATP. And a different little organism, now called a mitochondrium, also lives in a eukaryotic cell. It also had the same little ATP synthase, a different mechanism to set up a gradient of protons that is being driven based on the breakdown of glucose, right? So the NADPH is being provided from the breakdown of glucose. So hopefully this is helpful and helps you with the in-class exercise and understanding the biology of photosynthesis and cellular respiration a little bit more.